Okay. So we're very lucky today to have Oliver Warrington, who will be talking about his PhD work. Oliver did his first degree in biochemistry. Yeah, biochemistry. Transitioned into cognitive neuroscience via the UCL master's course, and he's just a few weeks ago. He's got your correction sorted for his PhD thesis, which he worked with Peter Koch and Phil UCL. That's the work that you're talking about today. So thank you, Oliver, for coming. Over to you.
Good. Thank you very much. Good start. So thank you everyone for coming on. That's all on my team. Thank you everyone for coming on, and it's very nice to be here. This is actually my first time ever in Cambridge, despite being from the UK. So it's a good excuse to wander around and have a look. So today, I'm going to be talking about my PhD work, which has been mainly focused on trying to determine the direction of communication between the hippocampus and neocortex with less specific fMRI. And I'm going to give one of those introductory apologies, because this is not going to be the smoothest talk in the world. I did just finish my thesis stuff last week, and the adrenaline that had been carrying me through for the last year or so has vanished, and now I'm just very tired. So I'll be taking a week off to sleep after this, but please excuse me while I try and stumble my way through the talk. So I'm going to give a little bit of motivation into why I wanted to do this in the first place, so a bit of a cognitive reasoning behind it, and then talk a little bit about the practical things that came up while we were trying to do this method, and then one of the results from my SMT studies. And for me, the most interesting thing, generally, but especially in cognitive neuroscience, is the fact that we can have the same stimulus, so the same sensory input, but our perception of that stimulus can change. So you may have seen these Mooney images before. The first time you look at them, it's pretty difficult to figure out what's going on. But then when I point out that there's a dog here at the front and the base of a tree here at the back, usually immediately becomes clear to people, and your perception of that image changes to the point where it's difficult to go back to seeing it the way you did before. So this, to me, says that our perception is not just the sensory input coming in, there's also an internal element, there's prior knowledge that we need in order to interpret that sensory information. And my very scientifically worded question that interests me the most is, where's that internal stuff come from? What's going on in the brain that we can have the same input with different experiences of it? And it leads to some very fun philosophical questions, like how can we trust the world around us? How do we know it's real? That kind of thing. But for me, jumping ahead, I just want to focus on the neural mechanisms that allow prior knowledge to interact with that incoming sensory information. So thankfully, the world around us is very structured. There's lots of things we can take advantage of and learn in order to have this prior knowledge perception. So even these arbitrary associations between two stimuli, you hear a low tone and frequently see this orientation of grading. You can associate those together to the point where you can start to implicitly or explicitly generate expectations. But once you hear that tone, you're going to see this grading. And the idea in perception is that you can have this top-down expectation signal interacting with your bottom-up sensory information. But to me, it just sounds an awful lot like memory too. You have a cued recall process, you hear an auto cue, have some time completion in order to retrieve the expected stimulus from memory. And so it makes sense to me, at least, that the hippocampus is involved in online perception, and potentially its role is to try and generate these top-down predictions to send down the cortical hierarchy. So just on the side, saying memory and perception have been linked. And I'm interested in the idea that there may be some shared mechanisms between what we see in episodic memory, where sensory information comes into the hippocampus, it does pan completion, and then there's causal reinstatement, that completed target. And this is an ideally suited mechanism for predictions and perception too. But with typical fMRI studies, we always struggle because we can see connectivity between the hippocampus and neocortex. There's other studies showing that the hippocampus is involved in predictions and represents stimuli, predictive stimuli. But we can never tell which direction that information is going in. It could be that the cortex is sending these predictions to the hippocampus, or what we suspect it might be that the hippocampus is sending these predictions to the cortex. So that's the question I tried to focus in on. And to answer the question, we are using layer fMRI. So with layer fMRI, the idea is that you can have high resolution fMRI. So at 7G, we can push the spatial resolution down to some millimeter levels. And this gives us the ability to separate signals from different layers of different cortical depths. And we can't see the exact laminar structure of the cortex, so we won't be able to separate 6 cortical layers. But generally, people assign each voxel to superficial, middle, or deep layer. And this is based on how much of that voxel's volume is in that particular layer. And usually people set some arbitrary threshold, say if more than half of the voxel is in the deep layer, then that's going to be called the deep layer voxel, even though you will get some mixing of signals from because of partial volume from the other layers. But being able to separate the layers of the cortex then allows us to use the known anatomical connections in order to infer the directionality that we otherwise wouldn't be able to do. So we know that the neocortex sends information feed forward through the superficial layers, and this eventually reaches the hippocampus, where it can go forward to the hippocampus through two different pathways. So, and then eventually it comes out to the neocortex again, to the deep layers. So it's a very simple idea. If we see something superficial, then it's going to the hippocampus. If we see something deep, then it's going from the hippocampus to the cortex. And this idea was based on 2 very cool studies that have used this, looking at encoding and retrieval in memory, where they saw that the hippocampus was sending information to the superficial layers from entorhinal cortex and to the deep layers from entorhinal cortex during retrieval, and the encoding was going through the superficial layers onto the hippocampus. So when I started my PhD, I was like, that's super cool, I'm going to do that. Looks fairly simple. It turns out it's not. It was a bit of a pain. And the first issue was that scanning the medial temporal lobe at 7T for functional scanning is very difficult. Typically, entorhinal just disappears. And that's the first problem we needed to solve. We were focusing in on entorhinal, and we wanted to be able to get to a point where we could say, okay, we have enough signal now that we would be able to be confident that we would detect a less specific difference if there was one. So the issue with entorhinal is that there's a lot of inhomogeneity in the V0 field there. So typically in V1, everything's nice and homogeneous, and you can set your protocol for the scanner to selectively excite water and suppress fat, and then everything's okay. You get the good signal that you're hoping for. Hippocampus and orbitofrontal cortex, they get a little bit worse. There's more inhomogeneity, which causes more dropout and distortion. And then you get to entorhinal cortex, and everything is just a mess. And there's a lot of negative V0 and a lot of positive V0, so you might try to address one aspect of it and then find you're losing out on a different portion of entorhinal. So the whole first year of my PhD really was trying to find this sweet spot where we could recover enough signal from entorhinal and as much of the whole of entorhinal as possible, and then just one section of it. And not only that, but we also wanted visual cortex ROIs, because these predictions are supposed to go from the hippocampus to the sensory cortex, so we also wanted to look at that. So we set our physics team a very difficult challenge. Essentially, we were saying, can we look at entorhinal in a very specific way, and the visual cortex? And there's a ton of stuff that we played around with. I learned that MR physics is just this huge parameter space where you change one thing, it affects another one, and you just have to juggle around until you find something that works. I'm not going to go into a ton of detail about it because I'm not an MR physicist, but two of the main changes that really helped us were changing the excitation pulse. So going from that nice water selective pulse to something with a bit more of a broad excitation profile, which usually you don't want because it might excite some fat that you're trying to suppress. But in this case, because of the broad E0 distribution in NTRL, you actually get able to excite more and recover more signal there. And we also had to drop the spatial resolution just to boost our TSNR. And typically, the layer studies we've done in the lab have been at 0.8 millimeter isotropy, but we had to shift down to about 0.9, I think. And this was based on another study from Essen, where they were trying to look at internal layers too, and they were able to find this triple dissociation in the layers. So this gives enough confidence that if we matched their spatial resolution and we were getting enough signal from internal, then we could go forward with the study. Like I said, there was a lot of other stuff that we tried out too, and the full picture and the full protocol that we ended up with has a lot of differences to the standard one we use. If anybody's interested, feel free to chat to me about it. I'm happy to, or at least direct you to the people who really did all of this and who really know what's going on. This is part of our physics team, and Nadina Gradle, the postdoc that really helps me, and Martina, the head of the physics team. So just to illustrate the kind of difference we ended up with, this was with 0.8 millimeter fMRI EPI protocol that was doing the fMRI in the medium prefrontal cortex. So already an advanced protocol that had been optimized from a different region. But in entorhinal cortex, we were getting much lower TSNR than our other regions of interest. And we had this usable voxels cut off where anything below 5 TSNR we would just consider as dropout. And in entorhinal, we were barely getting, we're getting just under 60% of the voxels passing that threshold, whereas our other regions of interest, almost every voxel is usable. And in the end, after that year of piloting and trying things out, we got quite a nice boost in the TSNR entorhinal cortex. And importantly, we got many, many more of those voxels to be usable. And I guess that's it. So this is the TSNR for the study that I ended up doing straight afterwards with about 30 participants. So every dot is a participant TSNR. So on the Y, we have the voxels that we consider usable, so above that 5 TSNR threshold. And on the X is the average TSNR of those usable voxels. And you can see entorhinal cortex and periorhinal cortex show quite a lot of individual variation still. There's still some subjects who have quite substantial dropout there. But if we consider this dot to be the starting point that we had from the piloting, we managed to get the majority much higher up and gave us more confidence in our study going forward. Ideally, one day we get it to look like periapical cortex or hippocampus, where we can use all the voxels and everything's nice and easy, but plenty of work still to do there. And just to say, for anybody else who's interested in doing this, one thing we struggled with is not being able to get a measure of the bulk response or the contrast signal to noise ratio in entorhinal reliably for a single participant, meaning we had to base all of our decisions on the TSNR, which you can improve without improving your contrast or even harming your contrast to noise ratio. So there's an element of faith involved that once you've got to this better signal point, hopefully it will just work for the bulk. So yeah, that's my very broad overview of the physics. But moving on to the actual experiment that we did and how we wanted to look at this prediction signal in the direction it's going in between the hippocampus and cortex. So the task here, we have auditory cues that are associated with visual shapes. And on a normal trial, a stimulus trial, so 75% of them, you hear that auditory cue, it predicts this first shape that is going to be presented. And then the task is to tell whether the second shape that participants see afterwards is the same or different to the first. The shape discrimination task is just to maintain people's attention, make sure they're looking at the shapes, but nothing to do with the expectation that we'll end up looking at. So there's no behavioral element to our study. Really, we're just interested in the effect of the cue and predicting the stimulus. And the most important trials for us are these emission trials, where we play that auditory cue and then we completely omit the expected stimulus. So the idea there is that we generate this top-down prediction signal by playing the cue, but then we isolate it from bottom-up sensory information by omitting the stimulus. Because when you're trying to look at feedback in layers, if you have bottom-up sensory information going too, then very quickly everything can spread across all of the layers, and it becomes difficult to sort of isolate what the feedback element of that is and what's the feed-forward processing element of it. And really, we're just interested in this top-down prediction and trying to figure out which direction that's going. So from here on out, I'm only going to talk about the emission trials. We use a shape-only localizer run in order to, as an independent run, to generate a shape-specific element of information when the shapes were not predicted, but they were presented, so that we can then compare that to when the shapes were not presented, but they were predicted. Hopefully that is going to make a bit more sense here. So what I mean by shape-specific pattern of activation. So we take the condition betas for each shape when it was presented in the localizer. So no predictions involved here. We do a subtraction between the shape 2, I call it, and shape 4. So shape 2 and shape 4. And the idea of this subtraction is that we remove anything that is common between those two patterns, and we should be left with activation that is just specific to the difference between these patterns. So that's why we call it shape-specific pattern of activity. And that's what we end up with here. We do the same thing for predicted but omitted shapes. So when shape 2 was predicted but omitted, and we subtract shape 4 when it was predicted but omitted, to end up with that same shape-specific pattern here. And it's often easier, I think, to think of these as like templates. So we're just correlating the similarity between these two shape-specific templates, one when the shape was presented, one when it was predicted but omitted. Does that make sense? I feel like I rambled. Okay, cool. So with this analysis, we're not addressing the directionality yet. We're just looking at which of our regions of interest contained information about the predicted but omitted shapes. And we first looked at the hippocampus as a whole, and we found this trend for a negative correlation between the presented shapes and the predicted but omitted shapes. And this negative correlation surprised us. We expected it to be positive. You'd expect the prediction of a shape to be similar to the shape when it was presented, or at least I would. So in order to try and figure out what was happening with this negative correlation, we went to a within versus between correlation instead. So if we go back here, within is just no subtraction, we're correlating within the same shape in the two conditions, and between is between the different shapes in the condition. So this would tell us whether the subtraction that we're doing is introducing anything weird that we weren't expecting. And it's a significant negative correlation in this condition. So it is information about the predicted shape that we're seeing in this analysis. But again, it being negative is surprising. So we don't really have a full explanation for that, and I can give you some speculations about why. But it is reliable. So in posterior hippocampus, we also saw this same pattern with a significant negative correlation from within. We didn't see anything in anterior, so from here on out, we just left anterior hippocampus, and we don't look at the subfields there. but in the subfields as a whole. So it's a little bit weird we left out anterior specifically, but we do include anterior when we look at the whole subfield. And when we look at the whole subfield, we see that it's just CA23 and the preparaceviculum that has this predicted but omitted shape information in it. And based on the hypothesis, this kind of makes sense because CA23 does the panning completion. So if you have that auditory cue, it would make sense for CA23 to panning complete the expected shape and retrieve that from memory in order to use it as a prediction. And then preparaceviculum is one of the major output subfields of the hippocampus. So that would also make sense if it was going from CA23 through the preparaceviculum and then out to the cortex. But of course, you can't say anything about that yet. It is surprising though, if you have CA23, you'd expect information to then go to CA1 and then out of the hippocampus. But we don't see any predictive or omitted information, shape information in CA1 or the ceviculum. And in the posterior hippocampal subfield, CA23 just start to drop off a little bit. We see a bit more of the ceviculum engagement, and preparaceviculum is really the most robust, I want to say site, just predictive omitted shape information. So we also looked in our cortical regions of interest. So we looked at entorhinal, perirhinal, and parahippocampal cortex, and then also LOC. And another surprising result is that we don't see anything in entorhinal or perirhinal cortex. We fully expected these predictions to go hippocampus, entorhinal, perirhinal, because that's supposed to be more of the object sort of pathway. Whereas parahippocampal cortex is the only one that does contain this predictive or omitted shape information. So that's true when we look at the whole ROI, but also when we split it into the deep, middle, and superficial layers. It's significant here in deep and not superficial, but you can see there's really no difference between these. So we can't say anything particularly less specific at this point, but we can say that there is this information in the parahippocampal cortex and definitely in the deep layers. So my speculation about why we might have a negative correlation, and if anybody has an idea, please feel free to share it with me, because I'll take anything at this point. But one idea is that the hippocampus is, the effect of predictions is supposed to be inhibitory. So if we look at predictive coding, solid worlds, then predictions are supposed to explain away sensory information, and therefore it would make sense if these predictions had an inhibitory effect. And although we can't look at excitation and inhibition in fMRI, this may appear as a negative correlation as the voxels that cared about the information when it was presented, and now having reduced activity when it's predicted. Does that make sense? So it may look as though it's like a negative image of, a negative version of the image when it was presented. Whereas with memory, you want to excite the cortex and reinstate that information, so that you're more inclined to have disinhibition as an effect of the hippocampus sending information to the cortex. So it would be interesting to look at whether there's any differences between predicted information and just recalled information. Another idea is that it's a negative prediction error. So typical prediction errors, you have a prediction and then there's some sensory information that wasn't predicted, and therefore you feed forward this extra information that you need to explain away. Whereas in our case, we have information that was predicted, but then no sensory input coming in. So in that case, you want a signal that tells the top-down prediction to stop happening. So that may be what we're seeing here, rather than a prediction. We might be seeing a negative prediction error, which I think would completely change the interpretation and lead to some other interesting questions. And yeah, we've stuck with the prediction because there's shape information that's just a result of the cue, but it's not conclusive, essentially. That's the point I've reached, I think. But just pushing that under the rug, essentially, and to finally get to the directionality question, we did an informational connectivity analysis. So here we take this shape-specific template that we get from the localizer, and this time we correlate it with single trial data from the emission trials. And this generates a time course of shape-specific information for each region of interest. And then correlating those time courses between regions of interest can give you an idea of whether they're sharing information between each other. So it's like a multi-voxel version of typical functional connectivity. And the main result of my PhD so far is that we see this connectivity between CA23 and the deep layers of the parahippocampal cortex, but not with the superficial layers. So based on our inference process, where we use the anatomical knowledge to figure out the direction, this would tell us that predictions are going from the hippocampus and to the cortex. We do also see with the parahippocampal cortex and the posterior, but also the whole prefrontal, there's connectivity across all layers of the cortex. So this may indicate that, speculatively, some of this deep layer connectivity may be a prediction going from CA23, prefrontal and then out to the parahippocampal cortex. But seeing superficial and middle layer connectivity there does suggest that there's also a feed-forward flow of information between the parahippocampal cortex and prefrontal. And why exactly that might be is unclear. We know that the prefrontal is a major source, a major target of visual input into the hippocampus. There was no shaped visual input on these trials, so that doesn't fully explain it nicely, I don't think. But there's the idea of the entorhinal cortex forming this loop where hippocampal output is then fed back as input, and it's not thought to extend to the parahippocampal cortex, but maybe it could. So as you can see, I think these last studies in the medial temporal lobe are nice in theory, but there's a lot of caveats to how we can interpret them. And if this was a conference talk, I'd typically have this summary where we saw predicted but omitted shape information in the hippocampus, the CA23 prefrontal and parahippocampal cortex. And then we saw the prediction being sent to the parahippocampal cortex and from CA23 and this bidirectional connectivity with prefrontal. Kind of leave it there. But because this is a method talk, I feel like I can vent a little about all my caveats and ask the question of, is that really what we're seeing? And one of my main issues with laminar fMRI at the moment is we tend to think deep layers is feedback and superficial layers is feed forward. But we know that that's not a nice split that we have in the cortex. There's a feedback and feed forward pathway in the superficial and deep layers. And so interpreting deep layers as feedback can be problematic in my mind. And the way we split the cortex up in the fMRI side of things doesn't match onto the anatomical side. And we can end up with layer 1, which is a major target of feedback, being mixed in with layers 2 and 3. And because it's such a thin cell-free layer, the contribution of layer 1 signal to that overall layer profile that we see may be just washed out by everything else, and therefore some of the feedback that we think is occurring, we might just not be able to detect. And yeah, I think that's a good flower. The other issue I have is with these pairwise connectivity measures we've been using, the informational connectivity. It could be that there's a common input, so some confounding region that is talking to both of the parahippocampal cortex and the hippocampus, and that explains everything that we might see connectivity-wise between the two. And one idea that I've been enjoying recently is the shallow brain hypothesis, where the subcortex, we know it projects to all regions of the cortex, and we don't really take that into account when we're looking at these hierarchical cortical models, particularly in predictive coding. But there's a lot of information there that gets sent to all layers that may explain what we're seeing here. And without looking specifically at these regions to see whether they have any predicted or omitted shape information in them, we might not be able to rule that out as an explanation. And again, even if we do look at those regions, maybe they don't pass the statistical threshold saying there's that omitted information in there. But when we're looking at these multivocal patterns, these threshold effects further down the line could be correlated, if that makes sense. So the connectivity may be correlated, even though the information doesn't pass the threshold. Hopefully that makes some sense. And yeah, as I mentioned, we can't look at that excitation and inhibition. We know there's inhibitory interneurons in every layer of the cortex, and since the bold signal is typically more reflective of the input to a region, that input may target these inhibitory interneurons, and the effect of that may be completely different to the effects that we think it's having. So as I mentioned, with the different explanations where predictions are supposed to be inhibitory and memory is supposed to be excitatory, we wouldn't actually be able to tell the difference with the method we're using alone. Likewise, there's a lot of local processing that happens in every cortical column, and fMRI is over such a long time scale that we're not just capturing that initial input or output of the region, we're also capturing everything else that's happening in between. So again, I think there's a lot of questions, and I think it's very cool to do, but by itself, maybe not very useful. I think with other methods along with it, like animal studies, or perhaps some more, like if we can do DCM and try and make some of these assumptions explicit, that might help the inferential process. And one thing I think would be very cool to do is to combine it with diffusion MRI, and I think Marshall Dalton from Australia has used diffusion MRI to find the endpoints of each cortical tool's connection to the hippocampus. So if we could combine that tract weighted measure into our connectivity measure here, that may give us more confidence in the anatomical side of this equation. So thank you very much. You'll be very patient. It was a bit strenuous. But if you are interested, there's a preprint out, and feel free to ask me anything both here and by email or whatever. And yeah, thank you very much.
Thank you, Oliver. Do you have any questions, Patty?
I wanted to ask about your thoughts on what those omission trials end up signaling, because they are all trials, like 25% the trials, and but when do people realize it's a omission trial, when it moves to the next trial, or is there like a fixation that signals that there's no stimuli coming?
Yeah, so we tell participants beforehand that there will be trials where the shapes are not shown, and their task is to just sit there and wait for the next trial, and just make sure they're focusing on their fixation dots still. So nothing happens on the screen except a fixation, and eventually the next trial starts with the auditory cue. In terms of what's happening on those trials, I'm not 100% sure. There's often debates when we talk about this as a group of whether they are predictions or prediction errors. You can argue that because it happens 25% of the time, it's like an oddball,
Yes.
and therefore there would be some surprise element to it. And the brain should be updating based off that prediction error that the thing it was expecting didn't happen. So I think that's a very reasonable account. And one of the issues with predictive coding is that it's very difficult to tell the difference between the two in our experimental measures. The prediction errors have stimulus information, the predictions are stimulus information, and all of it's supposed to eventually cancel out until you've settled on your representation of that stimulus. So in all cases, there's stimulus information that we're trying to detect. And because we don't have the time resolution with fMRI, we can't say what that initial input and output was. And with MEG reader or EEG, we don't have the spatial resolution. So it's a little bit like, how do we do it? How do we figure out what's what? So at the moment, I'm just saying whatever you like to interpret it as. It's completely fine by me.
So you skip a lot of details for the acquisition in itself, but from what you were saying, assuming it was a 3D EPI acquisition?
Yeah, that's right, 3D EPI.
Okay, and you had quite a large volume coverage. So what was your TR?
So TR was 3.43, I think,
Okay,
seconds.
so just under 3.5 seconds
Mhm.
for a 3D acquisition. Are
Yeah.
you worried about movement at all?
So generally always yes. But for this study, we had, we call it Harold, I don't know what their official name is, but pads around the participant's head that you can blow up and sort of reduce that range of movement that's possible in the scanner to a comfortable point, of course. And we found that that was really helpful in this study. I saw a lot less movement here than I had in the previous MG study. And generally we exclude participants if threshold, if they move more than a millimeter in like a single jump 10 times, then we exclude them. If they move so that the field of view is shifted, I can't remember the exact threshold, but past a certain amount between runs, then we also exclude them. So we try and have a strict criteria that lets us be more confident that we're not just affected by movement.
And what was your exclusion rate?
So I scanned 40 participants, 30 ended up in the study, and I think 5 were excluded from movement.
And but so 3D acquisitions are particularly valuable for within volume physiological noise.
Mhm.
And obviously the padding is not going to help you with at all. Did you collect any extra information, respiratory belts, these sorts of things?
We didn't for the study, which I think is important to do for next studies, because I think you become even more sensitive to this stuff with this 73D UPI things. And we did try other methods. Aroma, I don't know if that rings a bell for anybody. Like an ICA sort of method to try and get rid of some of that noise. But we found that it didn't actually have any effect on the results that we were seeing. So instead of making the pipeline more complicated, we just left it up and it was a more simple thing. But I do think it would be very useful to get rid of those physical aspects of them. I know we did a segmented acquisition, which helps with that within volume time, so you don't get as much distortion, which potentially helps us with the physiological side of things too. And in our pre-processing, we also used Nordic to try and get rid of some of the thermal noise on these images. So it's the best we could do, wasn't it?
And so your segmented acquisition, was it 2 folds or
Yeah.
2?
Yeah. We did try 4, and we saw it was better for distortion than that. GR just becomes too long, not feasible.
Thank you.
Yeah, I was wondering if you could go into a bit more detail about the TSM calculator that you used, because it was very small on the slide, so I couldn't actually see what the criterion was.
Yeah, so any voxel with TSM lower than 5, essentially. And this was based on us doing a lot of studies in visual cortex, where TSM normally usually a lot higher than that. When I've looked at some of the other fMRI studies that looked at internal cortex, some average TSM is actually around 5. So they would be including a lot more voxels that we've excluded. I think that's not ideal because it's such a difficult region to scan and with such low signal. And if you're looking at something that's predicted but not presented on the screen, you're just accumulating chances to not find anything.
And for the delineation of the MTL subregions, did you use FreeCipher?
No, so we've used FreeCipher to get the surfaces that then turn into layers further down the pipeline. For the MTL ROIs, we use ashes. And Alex Hickley, who's in the department, has segmented, manually segmented a bunch of the same 70 data that I had, and turned it into an ashes atlas. So thankfully I didn't have to do any manual
Is that
segmentation.
publicly available?
Yeah, it's published now, and I think it's available to download somewhere.
So that includes like the pre and paracepiculum as a separate region?
Yeah, and so that atlas does just the hippocampus subfields. The MTL cortical ROIs I got from a different atlas, which was a 3D atlas done from Princeton. And that's the same atlas we've used in previous studies.
Okay, so you used ashes not for the MTL cortex?
We used ashes for the hippocampus subfield and the MTL cortex, and then FreeCipher for everything else.
So, got a couple of questions, that's right. Asked a question about the emission response. So if you do MEG, EEG on these kind of tasks, there is a big response locked to when in time the stimulus should have occurred, which is pretty more like an emission error rather than prediction. And so when Ian Iyink was here, he did a paradigm very similar to yours with MEG. The idea that we can separate temporally the prediction period for stimulus occurs without having to have emissions. But we found it actually surprisingly hard to decode any predictions. So we could decode in a localizer the orientation of the gratings, the visual gratings, but we couldn't decode locked and auditory stimulus, the orientation at all. And so I'm wondering how strong the kind of visual predictions really are in this paradigm. People really report like by seeing a grating when they hear the rising tones or whatever, because in our case, participants, they could tell you explicitly, oh, I know this tone is going to be followed by this orientation and this, but they didn't seem to be having any visual predictions that we could decode with. Yes, for the orientation of MEG, but the fact is we could decode it with non-screen, but just the predictions seem to be very weak, and we never followed it for that reason. So I guess I'm wondering really how confident you are that there is some kind of prediction going on, at least it's a visual prediction.
Yeah, it's a very good question. So
if you had a behavioural thing, right, some kind of fast reaction times.
Yeah, it would be nice. And we tend to ask participants after the scan, we are aware of any relationship between the cue and shape. And most people just say they didn't pay attention to the reviews at all.
Okay.
We do explicitly tell them beforehand this cue is a relation, but it's not important for the task, and they're in the scanner for an hour and a half.
So they're not, so our participants are actually told to try and learn these, in fact, we give them a training period to do that. And so maybe because we're doing it explicitly, where you do more implicitly, maybe that's.
We do have another study doing exactly the same, where we're tracking learning over time, and we're hoping there might be a switch of direction. So when you're learning, it's more about prediction errors going forward, and when you've learned, it's more about predictions going back. And in the 3-2 version of that study, you can see the prediction representation switch halfway through. Whether that will be detectable at 70 and stuff, not so sure. And I think the prediction that we're talking about here is a very implicit centric processing sort of prediction rather than I know what's going to happen. So,
okay, we making explicit people were overshadowing our study, they were explicitly verbalizing and not actually using
which is a little bit challenging because the predictions we tend to talk about in predictive coding and not even these sort of temporal predictions that we use in the studies. So how best to kind of get at that predictive element, isn't it?
Yes. Second question, one of the caveats you didn't mention a general caveat with bulb, of course, is the superficial bias, that the draining veins go from deep to shallow and will tend to merge signal across layers, and there'll be a higher SNR than superficial layers, which I think applies to 3D. But that's interesting because you actually found the quantity in deep layers there should be worse SNR rather than better. So I guess your results are not trivially explained by bias, high SNR in
Yeah.
the regions of deep regions, which I guess is good news.
Yeah.
Because I said that I mentioned earlier that paper with Johan simulated even patch analysis still sensitive to more explicative SNR. So even if you're doing path analysis, it's still sensitive, it's biased, but here it can't explain your result.
Yeah.
It's most of it.
Yeah, I think we definitely have knowledge of bias in this, and we're just very happy whenever we see deep and not superficial effect, because then we can just ignore it and say it's not influenced at all.
Yeah.
So,
Final question, this idea that the negative correlation you're getting is because you've got your predictions as inhibitory, so inhibiting the thing itself, the negative pattern. And I guess you could expect that if it's like in paracampal gyrus, the hippocampus was doing the prediction inhibiting, but you found that in the hippocampus itself as well.
Yeah.
Negative correlation is posterior.
Mhm.
So is that hippocampus inhibiting itself in some kind of loop going around? I mean, when you think of pattern completion, given the tones, you normally think that somewhere does reinstate the actual pattern, which may inhibit lower areas.
Yeah.
How's it inhibiting itself? How do you have pattern completion and inhibition in the same?
Yeah, that's a very good point, and I think that's one of the reasons I suspect there may be some common influence. It would make sense to me if, like, the medial prefrontal cortex was inhibiting the hippocampus, for example, and also potentially hippocampal cortex. We have tried to do a search slide, so we do the same pattern similarity analysis across the whole slab. We don't see the whole brain, we get like a flat slab like that. And for the emissions, nothing happens multiple corrections, not cluster correction. But we do see, when corrected, there is a small blob, and just in the orbital frontal cortex. You remember anatomy is not fantastic. And prefrontal hippocampal cortex is also small in the cerebellum. So I think there's maybe something frontal cortex going on, maybe some timing element from the cerebellum going on.
That'd be very exciting. I mean, I would also expect medial frontal cortex to inhibit hippocampus. There's some evidence for that, so it'd be amazing if you did find that.
Yeah.
Did you just not do an ROI just to avoid the problems of comparisons? To say, let's take an ROI about the people like Eleanor have in the past and treat that as another ROI and see whether you get negative, positive patterns.
Yeah.
Or maybe the sequence is not awful. But also suffers from dropout.
Yeah. We definitely could.
Right.
And I would like to. I think in the future, I don't know if we'll be able to look at the layers there.
I don't know.
Because we've optimized for a different region, but that would also be really cool if we could.
Yeah.
Follow up on the inhibition part. Okay. This might be a stupid question, but why would we expect them to inhibit? Because the task doesn't require them to, it doesn't help them to inhibit it, right? And it's also, we also don't know when they realize that this is an omission trial and they decide to inhibit as well.
Yeah.
So why would there be an inhibition is my question, I guess. Because it's the task, it doesn't help with the task, they don't need to respond anyways. So yeah, I was confused by it. Doesn't make sense.
Yeah, that makes sense. Yeah. I think the inhibition we're talking about here is very like automatic. You've got a perceptual process going on where predictions are constantly being sent down the cortex to explain away the input that we can predict already. And whether they predict or inhibit is very like subpersonal, like just neural stuff.
I see.
So the inhibitory predictions comes from predictive coding where it's supposed to explain away. That doesn't have to be the only case. There's other ways to formulate predictive coding where predictions go up the cortical hierarchy, for example, and those alternative explanations are not given much time. So yeah, but that's well, the general idea of inhibition comes from.
So the classic one is the extracellular receptor fields. We have a simple neuron B1 responds to a pointed bar, but if that's part of a larger bar goes across the visual field, you actually get no firing anymore of the simple cells in that bar, just at the edge, and it's not predicted. So yes, this idea of predicting away, explaining away is quite a strong, quite well grounded in physiology. Any more questions?
Yeah, I'm just very curious about where exactly in the cerebellum you see those effects.
Good question. Excuse me. It's my thesis. I think actually, it's a very rough and ready sort of correction style image, but the emission is the blue that we see. So there's that small prefrontal cortex of prefrontal region, and that's the small cerebellum blob that we see. I don't know anything about cerebellum, so I don't know what.
I wouldn't expect this more posterior, but it's interesting for sure.
Okay.
Because there's some really interesting hippocampal cerebellar interactions as well. And they show up all the time in memory tasks that I've done with fMRI as well.
Yeah.
So
Okay, cool. Yeah, I only know cerebellum plays a bit of a role in timing.
They say in the
Yeah.
context of the pain, so.
Yeah. And I didn't show any of this, but we did also look at those stimulus trials where the predicted shape does show up. And in case anybody's interested, we only see visual cortex representations there. We don't see anything in the medial temporal lobe anymore.
Okay, well, let's thank Oliver again. Oliver is around for